Dobry wieczór Państwu. Witam w kolejnym dniu festiwalu Kopernika. Wykład będzie w języku angielskim, ale zanim do języka angielskiego przejdziemy, to musimy Państwu odczytać takie informacje na temat naszych partnerów i sponsorów. Partnerem Copernicus Festival jest Małopolska Organizacja Turystyczna. Projekt jest dofinansowany ze środków budżetu państwa przyznanych przez ministra nauki i szkolnictwa wyższego. Dofinansowano ze środków ministra kultury i dziedzictwa narodowego pochodzących z Funduszu Promocji Kultury Państwowego Funduszu Celowego. Partnerem wydarzenia jest miasto Kraków. Copernicus Festival został objęty honorowym patronatem ministra nauki i szkolnictwa wyższego oraz prezydenta miasta Krakowa Aleksandra Miszalskiego. So, good evening everyone. It's a great honor and pleasure to welcome you here in Copernicus Festival 2025, the mystery. Today we are focusing at mysteries of mind. And my name is Krzysztof Cipora. And it's a great honor and pleasure to introduce Professor Stanislas Dean, who would give us a lecture about the past, the presence, and the future of brain imaging. Professor Dean is a professor of Collège de France and a director of Neurospin in Paris. And without further ado, Professor Dean, floor is yours. Thank you so much for accepting the invitation.
Hello, good evening. Thank you very much for welcoming me here. It's a wonderful pleasure to speak to you about the mysteries of the brain and the mysteries of the mind. This is the place where I come from in Paris, Neurospin, in the south of Paris, in Saclay University. And each of these beautiful arches that you see here is hosting one brain imaging machine. You know that with MRI, with MEG, with EEG, I will explain to you these acronyms, we can begin to unravel the mysteries of the human mind. And it's probably the most complex objects in the universe. So it's a really exciting scientific endeavor. What we are after is illustrated in this actually quite old engraving from the 16th century, Matthias Greuter. And of course, with our modern eyes, it's impossible not to see someone being scanned in an alchemy oven, actually. And what's interesting is that what comes out of the alchemy oven in this engraving is not pictures of the brain, it's pictures of the mind. It's the ideas that the person has in mind. He's thinking of his mistress, maybe of a horse, of fantasy people. And that's the question I would like to ask. If we scan the brain, can we understand the mind? Can we shed light on our mental representations? Is it possible? Is there a direct link between the two? And I will show you that we are reaching a certain level of success. And I will show you how. Of course, it started a long time ago. For several millennia, people have known that there is a relation between the head and thought, or the brain and thought, but they had the wrong models of how it worked. This is the model that started with Aristotle and Galen and lasted until the Renaissance. It's, of course, every generation has its own metaphors for machines like the brain. And at that time, the machines were hydraulic machines or machines based on weights. And the idea here is that the machine of the brain is a hydraulic machine. It has several places where there is water flowing, and the flow of water is doing computations. That's actually not ridiculous because there are actually ventricles in the brain full of water. And so the idea of this time was that the first one would have the sensus communis, the common sense that brings together the information. You can see the wires from the nose, from the eyes, from the mouth, of the taste, and so on, brings it together. Then the second stage is cogitation, and back, appropriately in the back of the mind, there is memory. Okay, that was a model of the mind for about a millennia and a half. And how could we know better It was very difficult to study the brain. It's at the Renaissance that people started to study the brain again thanks to dissection. And Vesalius, for instance, produced these gorgeous images of the brain as well as the rest of the body. But it's not enough to look at the brain. It doesn't tell you how it works, right? You can see the beautiful organization. At that time, anatomists recognized some of the major sulci and gyri, the folds of the brain, but they did not know how it works. And you would have to wait until the 19th century for this particular brain. This brain is the brain of the very first patient with aphasia, the patient of Paul Broca, French neurologist, who jumped on the occasion. He had a patient who could no longer speak. He could only say tan, 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 tan. He could only say this, as well as Sacré nom de Dieu, holy name of God. It was an interjection, so interesting. Perhaps there are different pathways in the brain for things like that. But anyways, the patient died from another cause, and Broca rushed, took the brain of the patient, and then rushed to the academy because he had made a splendid discovery, which you can still see. This is the original anatomical piece, which is preserved in the Paris Museum. There is a lesion in the foot of the third frontal gyrus, and that is, Broca thought, the origin of the deficit, the seat of articulate language. And Broca was the first to recognize that there might be a lateralization to the brain, because this is in the left hemisphere, that this sort of lesion will cause aphasia, difficulties in producing articulate speech in this particular case. For a long time, this was the only method, correlating brain lesions with their consequences on the mind. I started with this before there was any imaging. We saw patients and we're trying to make inferences about how the brain worked from the dysfunction of breaking the machine. Imagine trying to understand these machines just by breaking them, right? That's not really easy. Then there was a revolution. And that revolution you see now, 87, 88, you know, some people have flashbulb memories. You all know where you were on September 11th. I know where I was when I read this paper, because for me it was something really extraordinary. I'm not joking. This was really a game changer. This is the paper by Michael Posner, Marcus Reichel, Steve Peterson, collaborators, where they were able to use a technique which is called positron emission tomography to see what different parts of the brain are doing in the living brain. These people are living. They are coming out of the machine alive, and yet we can see inside their skulls. And how does this work? You inject a small dose of radioactivity, small enough not to be dangerous, but enough to see where the radiation is coming from in the brain, and it's accumulating in places due to blood flow where the brain is actually working more. And so you could see that when you are reading words, I'll come back to that, you have blood flow in the left posterior part of the brain. When you are hearing, it's a different area, auditory areas of the brain, superior temporal lobe. When you are speaking, it's yet another motor region. But most importantly, when you are generating speech, verbs associating to nouns, you have activation in Broca's area. A purely cognitive function is associated with a separate territory of the brain. And this technique allowed to map any capacity in the brain. So we started to all use this, but it was very primitive. You had to have a cyclotron in the basement. So we had a little cyclotron in the basement. The chemist would generate labeled water with oxygen 15 instead of oxygen 16. And we would have to rush because the half-life was very short. And so we would rush in the corridor with a radioactive syringe, inject it to the happy participant who was there for 3 hours. And we could get one image. Actually, this is an average of several subjects. We had to average a lot. But we could only get one image of the brain every 15 minutes, 12 to 15 minutes. Very, very slow technique, and yet revolutionary. Suddenly, the brain-to-mind mapping was becoming visible in a living brain. But now this is today. This is, of course, functional MRI, magnetic resonance imaging. And that was yet another revolution. With MRI, there is no radioactivity. There is no injection. what we see is the contrast, which is just due to the blood flowing inside the body and the oxygen that is brought by the blood to places that work in the brain. And we can do images at an incredible pace, less than 1 second for the whole brain in many cases. So we can have all of these snapshots of brain activity. And this image is from a single person, no averaging necessary anymore. A single person, any of you in this room could come to one of our scanners and in a few minutes will show you your brain organization for language. The colors here are of course fake colors. The shape is also a bit fake because this is the normal shape and this has been inflated on the computer. We have all sorts of computer techniques to reconstruct the activation, see it better. And so this is an image from Jack Gallant in Berkeley showing, that's already a few years ago, it's like 9 years old now, and he was showing that when you are listening to a story, different words, different aspects of the meaning of the story activate different parts of the brain. And so, for instance, in this part, which is labeled in red, you have, this part will be sensitive to words that relate to psychology, to social relationships, words like wife, remarried, he confessed, husband, requested, mother, and so on. So words that evoke the social field. There's a social network in the brain. That's one of the discoveries of the past 15 years. But there is also a number network in green, like in dark green here or here. There is also a network for objects and so on and so forth. The brain has become parsed into all sorts of places that care about different aspects of meaning. And that's feasible now in the space of a few minutes, thanks to this revolution. Now here comes our latest machine. This is Neurospin, and you can see that you see in 2017 the arrival of this giant machine. Our field requires something like telescopes now. This machine is called ESOLT. It's a unique machine in the world. It weighs 150 tons. It's filled with liquid helium. It's a prototype, and it's a magnetic resonance imager, MRI, which will reach the field of 11.7 Tesla. Unprecedented magnetic field for humans. And it works. It took quite a while, but now this machine is producing its first images. And the reason to go higher in magnetic field is that we can make higher resolution images, such as this one, for instance. You can see many of us have 7 Tesla scanner. That's a unit of magnetic field. But here you can see images with the 11.7 scanner and see the level of detail. Maybe we can zoom in. And if I zoom in, you can see this incredible level of detail about the anatomy of the brain that we are getting, the layers of the cortex. It's a surface that's been folded, but it's actually made of thin layers, a bit like in archaeology. And we're trying to understand what the different layers are doing. And this is just anatomy, but we have never seen an anatomy of that level of contrast and resolution before. And it's not just anatomy. You have to understand that MRI is extremely flexible. With MRI, we can do all sorts of different images of the brain. This is an image where the colors do not represent activity of the brain, but they represent the wiring of the brain, the connections between areas. So the colors here represent the direction of the connections in what we call the white matter, but you see it's not white here, it's all colors. So the red here indicates connections in this direction. The blue is connections through the plane like this. And the green is connections in the last direction from front to back. So you can see that we are able to get a level of detailed resolution with these new scanners that is truly amazing. This is 200 microns resolution. This is the work of Cyril Poupon in Neurospin. It's one of the best atlas of a single person, actually post-mortem here. And we are still atlasing the brain. You have to understand that even to this day, 2025, we still don't have a perfect map of the human brain. We are still mapping. And there are still many terra incognita. But I go back to the function of the brain. What's wonderful with the scanners is we could take any of you in this room. Here there are 3 different people, okay, 3 different brains. And in the space of a few minutes, we can figure out what different parts of the cortex are responding to. In this experiment, we are showing pictures of bodies, faces, places such as houses or outdoors, objects, and of course, written words. And you can see that there is quite a bit of spatialization. In every one of your brains, there are regions that care about places, midline here in both hemispheres. But there is a region that cares about written words only in the left hemisphere, which is shown in the bottom here. And there are minuscule patches that care about faces or about bodies. They're all very specialized. We are still discovering the extent of this specialization, and I'll show you that it is in part the result of education and learning, and in part the results of genetics. But how can we validate these images? What do they really mean? It's important for you to understand that we are not really seeing neurons. We are seeing voxels. They are the equivalent in the volume of pixels that you are familiar with from your cameras. Voxels are little volume elements. Voxels will be about the size of 1 millimeter, roughly. They can be smaller or larger, we can choose. But that means that they still contain thousands of neurons. And we do not see single neurons with this technique. And that's a limitation that is very strong. We would love to see single neurons. Sometimes we can. And I'm showing you here cross-validation by the ability to record single neurons in rare cases of patients with epilepsy. When patients have epilepsy, sometimes they are explored with thin electrodes for therapeutic reasons in order to explore the origins of epilepsy. You put thin electrodes and you can record, and sometimes even from single neurons. And this is one case here. You see a single neuron. All of these panels, they're a single neuron responding to different pictures from Florian Morman in Bonn. And what do you see? You see that just like there is a patch of cortex that responds just to places, this neuron responds only to pictures of places. It can respond to the pyramids, the desert, what have you, the Golden Gate, but if there is no place, it does not respond. It codes the presence of a place on the screen. You all have neurons like that in your brain, okay? That's how you parse this room, for instance, the external world, and other neurons respond to faces. And their existence confirms what we see with fMRI. What we see with fMRI is a sort of average of neural activity from thousands of such neurons. So here is another neuron that was very spectacular from the work of Rodrigo Cuyan Quiroga. Those of you who are close enough, can you see what it's responding to? Do you guys know about Luke Skywalker? I hope so. I think everybody knows about Luke Skywalker, right? Well, if you do, you have a neuron like that in your brain. I forgot to say, when you listen to my talk, try to think, if it's possible, that this is going on in your brain at this very moment, right? So this is in your brain. There is a neuron, this was recorded by Rodrigo Cuyan Quiroga, that fires systematically. You see all of these spikes whenever the picture of Luke Skywalker appears on the screen. These are called concept cells, because they respond to a particular concept. This cell responds to the concept of the person, Luke Skywalker, even though the person does not exist, right? But you have a mental image of that person. It responds to the picture, regardless of the movie or the age. It responds to the name, Luke Skywalker, and it responds to the name when it's written or when it's spoken by a male voice or by a female voice. Okay. So this is a very, very abstract neuron. It's responding to a concept regardless of the modality of input. There is convergence to this particular concept. Can you see, so you can see that it's not responding to Emma Thompson or to Shrek, come back to that, or to Sylvester Stallone, but can you see who else it's responding to? Yoda. I heard Yoda. So yeah, so this is very, very funny. It shows you a little bit the limit of these experiments. So the experimenter presented a picture of Yoda, and yes, the neuron fired just as strongly. We don't know what it means. And these experiments are limited in time, and we have no further evidence. Maybe this neuron is responding to all Jedi. Maybe it's actually a Jedi concept neuron, right? It's possible. Maybe it's a neuron that responds to Luke Skywalker, but Yoda is enough to evoke Luke Skywalker. We don't know. But we are still working on these neurons to understand exactly what they do. Concept cells. Now, because there are scientists in the room, I always want to show some really exciting new hot stuff. And this is stuff in collaboration with Peter Roefsema and Doris Dijkster-Huys, I think, from Amsterdam. We did this work recently that was published in the journal Science, in which we exploited these concept cells to get to something even more striking, the evidence for pointers in the human brain. So here we start with something very simple. We have a neuron that's a concept cell. In this case, I'm not joking, this cell is responding to Shrek. Okay. So you have a neuron that responds very strongly to Shrek, and if you show a picture of other people, such as a family member from the patient, does not respond. But maybe another neuron responds to the family member. So would they also respond to pronouns and not just to nouns? Pronouns are very interesting, right? I can say a sentence such as Shrek and Courtney Love went into a bar, and it sounds a bit like a joke, right? But then I can say he sat at the table, and who sat at the table? Well, it has to be Shrek, right? Because it's a masculine, and Courtney Love is a girl. Okay, it's a woman. So in this case, it's very interesting because the pronoun he is pointing to this particular concept only by the fact that it's occurring in the appropriate text, one after the other. Okay. So would the neuron reactivate? The answer is yes. It's enough to present a single pronoun pointing to the concept for the cell to reactivate in your brain. So in this case, you see the neuron responding to Shrek, walked into a bar, and you see reactivation only when then the patient read he sat on the table, not when she sat at the table, or not when it's he sat at the table, the black line here, but there was no Shrek to begin with in the original sentence. Okay, so this is the result of the average of 53 neurons, and perhaps even more impressively here on the right, this is what happens when we decode what the patient was thinking. We can build decoders that start from brain activity and decide which of three possible nouns was the referent of the particular sentence the patient was reading. So here you see decoding, and some of you see this scale, it goes all the way to 1, which is 100% decoding. We have three possible choices. We are decoding from brain activity what the patient is thinking about. And so when the Shrek comes first in the sentence, or when it comes second in the sentence, does not matter, we can decode it, but then we decode it at the time where the pronoun is presented and it refers to it. In other words, there are not only concept cells in the brain, but there is a system of pointers, which in language are nouns, pronouns, indexicals, and so on. We can point to concepts and we can manipulate them in all sorts of ways. There is a symbolic machine in the human brain, and we are not yet understanding how it works, but that does not mean that it's not there, you know. Maybe we'll debate with Terry Sejnowski a little bit later on. There is a connectionist movement that at some points want to get rid of symbols. But I think symbols actually are characteristic of the human brain, and we just have to find the right machinery for how they work. And this is perhaps a small contribution to understanding how symbols work in the human brain. But we still have a long way to go. We don't really understand this aspect of the human machine, how symbols work. It's just the beginning. Okay, so a key aspect of the human brain is that it educates itself. I think this is perhaps the characteristic of the human brain. I wrote a book called How We Learn, where I try to explain that this is really the unique characteristic of the brain. This is our extraordinary ability to learn more than other animals because we educate ourselves. Okay, so can we see traces of that in our experiments? The first thing is to recognize that learning starts extremely early in the human brain. And there is, in fact, abilities that serve as a foundation for later learning that are present right from birth. So, this is one experiment where we showed this. It's actually more than 30 years ago. This is the very first work that I published together with my wife. And we had this technique that just emerged that allowed us to place little electrodes on the surface of the head. So this is the occasion to introduce you to another technique, electroencephalography, EEG. I think it's familiar to you as EEG, where we record the brain waves from the surface of the head. There is no need to go deep, but we can record the sequence of brain waves and understand something about the computation of the brain underneath. And in this case, the experiment is very, very simple. The baby is hearing a series of syllables, ba, 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 ga, okay? Or ba, 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 ba. And when you do this, the brain habituates. I hope you can see that the signal is going down. Starts very strong for the first syllable and then goes down. And when you change the syllable, the signal goes up again. Not in the first place, but a bit later in time. Okay. What does that mean? That means that the brain is capable of discriminating Ba versus Ga. Already at a very young age, and in fact experiments have been done in a few hours of life, babies are equipped with the ability to encode language and to discriminate syllables. That's part of our endowment. We start life with that. And it's extraordinary because it means that right in the first year of life, babies are already starting to learn the language. They're starting to converge onto the specific sounds, onto the specific words, syntax that is being used in their language, but they already have the equipment to discriminate essentially all of the syllables of all of the languages of the world. And they also have the appropriate brain areas. This is, again, work that my wife and I did in 2002. We were the first to do fMRI of little babies. Maybe during the discussion we can answer your questions. It's not dangerous. It can be done very safely. We were very precautious with these wonderful little babies. They were protected from the noise and so on. But we managed to have conditions where they are awake, they are listening, and they are listening to sentences in their mother tongue, okay, in their native language. And we were very surprised to see, and many of our colleagues were surprised to see, that it already converges to the appropriate network, the very same network that you would find in an adult. So you can see Broca's area. This is the left hemisphere. It's left lateralized, inferior frontal region, and all of the temporal, superior temporal, middle temporal regions, very similar to what you would find in an adult listening to speech. It was different when the babies were listening to music. And so the conclusion is that the brain is already very equipped, very structured, right from the start, to learn language, to learn other domains such as number. I'll come back to that. And is pre-wired in a certain sense to route the inputs to different areas depending on what is their content, right? That's how we start in life. And I say this because it means that we are social animals. We need language input. And in this beautiful image, it's nicely illustrated. This is an image by researcher Rebecca Sachs. Professor Rebecca Sachs is at MIT. By the way, I think we should say a little word about our colleagues from Boston, from MIT, from Harvard in particular, who are being attacked today in a way that is truly unequaled, I think, in the history, in the recent history of attacks against science. So I'm thinking of them in particular. Sorry for this bit of anxious thinking, but Harvard is being under incredible and totally inappropriate attack, and science is under attack. Well, this image was acquired next to Harvard at MIT, and Rebecca Sachs had a baby. She went in the scanner with her baby, and you see this beautiful slice where you see the social relationship being nicely illustrated. On the left is just an anatomical image, just for the beauty of it. But on the right, it's functional images. The top one showing you the language network already being activated at this age when you speak to your baby, and the bottom one showing you a result of that. This is work from John Gabrielli, Romeo et al., showing you that if you scan a 5-year-old, you see the impact of the acquisition of language. The more you speak to your baby, the more this language network develops, such as at 5 years, this is the correlation between the number of dialogues that the baby has been exposed to and the activation in the brain. I think there is a very important message here. The brain is a plastic organ in this age in particular, between 0 and 5. Of course, it continues, but it's a huge amount of plasticity. And if the brain is not being stimulated, these years will be lost. You know, it's not replaceable. We have a limited window of plasticity. And it is very important that our children are stimulated with school, but also by their parents. And I think there is an important message for society today, because unfortunately, I'm afraid, we all have these little devices and we're all like, oh, okay. And our babies, our children, may not be getting the same amount of stimulation that they were getting just a few years ago. Well, this is evidence, growing evidence, that even the first year of life, when they're not even speaking, is already a critical moment where children need to be spoken to, because they are registering essentially all of the aspects of their mother language. Now later, a bit later, you're going to learn to read. And in my lab, we did a lot of work on what happens when you learn to read. You change your brain by learning to read, but what changes exactly? So we did this experiment. They were really exciting experiments where we decided to compare illiterate people who had never been to school, just because there was no school or their parents were not able to send them to school, with people maybe from the very same family that had been to school. Okay. And to make this contrast so that we could map out all of the changes that occur when you learn to read. Okay. So this is your brain on reading. This is a view. Each of these color areas here represent areas of your brain that have been changed because you have learned to read. And this is in response to a written sentence that flashes on screen. Okay. So what do we see? So the areas in color here have more and more activation in people who can read better. What you see on the x-axis here is the number of words that you can read per minute, and on the y-axis the amount of activation. So the first thing we see is that reading gives you access to the very same areas that respond to spoken language. Because you've learned to read, spoken language is now available from vision. There is a new interface into spoken language. But in order to do that, the reading develops the visual areas of the brain as well as the spoken language areas of the brain. So the top one, it was actually a surprise. You know, here we have subjects listening to speech in the scanner, but the amount of activation in the planum temporale, the superior temporal region that responds to speech and phonemes, is almost doubled in people who have learned to read. Because you've learned to read, you've changed your visual system, you discriminate letters better, but also you've changed your phonological system, you discriminate phonemes better, you hear better the sounds of speech. And we also found anatomically, with diffusion tensor imaging, that reading reinforces the connection between the two. There's a stronger connection between these systems. So we think we understand quite well now what's happening with reading. We've also consolidated this with scanning of young children. This is also quite an adventure. So this is the lab, but this is actually not a scanner. It's just a piece of wood that helps us to train children to get in the scanner. We recruit young children from the age of 5, 6, 7, and they go in the scanner and they learn to be still and have the buttons, have the coil, have little headphones, and they are like little cosmonauts that help us explore the brain. We tell them actually that they are like pilots of a spaceship. And of course we tell them, if you move, don't move, because if you move, the spaceship will go astray and will go to the wrong star. So there's a whole story behind it. But eventually they become really helpful, and they love these experiments. So a few years ago, we got the permission to scan the same children many, many times. 6 times, plus another one that you don't see here later on. So every 2 months, the same children would come to the lab and they would be scanned. And the first 2 scans on the top left were before they enter school. And we chose children that had not learned to read at all. And they just learned to read in French schools, which were working, good. So if I zoom in, what you see here is the responses of what we call the visual word form area. The visual word form area is a region of the brain that develops its response to written letters, the letters that you can read because you have been educated, you've been to school, and you've learned to read. So your visual word form area responds to alphabetical letters, Roman letters. And if you have not learned Chinese, it does not respond to Chinese. If you have not learned Hebrew, it does not respond to Hebrew. It will only respond to the alphabets that you've learned to read. We've studied this, by the way, with Professor Marcin Sved, who is here in the first row. Did beautiful work comparing Chinese reading and alphabetical reading. So we started this together in Paris. It's nice to see you, Marcin. So here you can see that basically as soon as children enter school, you begin to see these very strong responses to alphabets that they can read. Okay. And it will grow and become very stable. And we think we understand quite well now what's going on with reading. Reading will specialize a part of your visual system that would normally respond to objects or to faces, but this part will become very specialized. I use the word recycled. It's being transformed into a system for recognizing letters. And it sends its output to the planum temporale, phonemes, and to other areas of the language system. And that's how we can convert little marks on paper into language, spoken language, just like if it was spoken language. Using our new scanners, this is at 7 Tesla in Neurospin, we can zoom in. That's one way we can use these new scanners. We can zoom in to a single person at millimetric resolution and try to understand what these areas are doing. And we're still making discoveries. This is our latest paper on reading, one of the latest. And this is one particular person. And we made a discovery, a bit like astronomers. You know, at the beginning of astronomy, they saw there were many stars up there, and then they were able to zoom in and see that they are not stars, they are galaxies. Millions of stars. Okay, well, it's the same here. We thought there was a visual word form area, and actually it's multiple patches of cortex, little spots of cortex that are super specialized for reading. And each of them responds very, very tightly, you can see on the left, to words. In this case, these are bilingual people. They can read English and French. And we could see that the very same patches of cortex respond to both languages because they use the same script, the same letters, right? But I don't know if you can see, but these patches of cortex will not even respond to numbers. They are ultra specialized. They are very similar shapes of letters and numbers, but because they serve different functions, our brains during education treat these stimuli as something very different. Numbers versus letters. So this area cares about letters, and as you can see here, it also cares about the statistics of the letters. So if you look at the top stimuli, they are the colors that you see in the bottom. On the top right, you have a French word. And you also have an English word. And then you have things that could be French words or English words. They are not, but they respect the statistics of the letters. Which letter could be next to which? And as you move to the left, the statistics violate more and more the statistics of French or English. Now, when I give this presentation, I joke that the one on the left could be Polish. So I don't know if it's true. Actually, maybe you could help me. So maybe for you, the statistics are sort of okay. I don't think so, actually. You understand the experiment, right? You violate the statistics of the language that you can read, and you can see with this tuning that, well, the early visual areas, they don't care. For them, it's just strokes, okay? They don't know about reading. They just know about little bars, and all of these stimuli contain little bars. But as you move towards these more specialized patches, you can see a very steep gradient, which says these areas really like the statistics of the language that you've learned. They like frequent letters, they like frequent combinations of letters. They have absorbed the statistics of the environment, the statistics of writing. Okay. And that's not a bad metaphor for what happens during learning. The brain is exposed and absorbs some statistics of the environment, or compiles some statistics of the environment. And that's why it takes time, of course, to learn to read, because you have to become specialized in all of these statistics. You have to see a lot of words, millions of words. We did the same experiment in Chinese. So here's a Chinese-English bilingual. And now the results are subtly different. Almost all of the reading circuit is the same for Chinese and for English. So in green, you can see areas that really respond the same to English and to Chinese. And they have the same sort of gradient. You can see, I mean, I don't think, maybe most of you don't read Chinese. I don't. So the characters on the right are real Chinese. This looks like Chinese, but it's not at all. And we break down Chinese in the same way that we break down English here. Well, you see that this patch of cortex likes both Chinese and English. This Chinese is English. But we made the discovery that there are little patches of cortex that care just about Chinese. Probably because the writing system is a little bit different. So the brain dedicates specific patches of cortex to this particular form of writing. This is a very recent discovery, and there's one thing we don't understand at all, is that the patches that like Chinese, they also like faces. They respond very strongly to faces, which is here. Okay. And we don't know why. There is something funny about that, the relation between faces and writing. Maybe Chinese characters are a bit like faces in their holistic appearance, but we don't know. We have more work to do. In order to understand what reading does to the brain, one great advantage that we have is that now we have simulations that are a little bit brain-like. And I'm sure we'll discuss that with Terry Sejnowski a little bit later on. We have neural networks, artificial neural networks that are made a little bit like the architecture of the brain. They have a series of stages. In this particular network, which has actually a good brain score, so it's not a bad match to the brain, we have actually labeled the different stages according to brain areas, as if it was primary visual, secondary visual area, etc., etc. And these networks simulate more or less how sets of neurons could recognize pictures. They are taught to recognize a face and to give a given output. But this is an illiterate network. So what we did in the lab is we looked at these networks by turning them into literate networks. We trained them to read, if you want. We gave them a lot of words at the input and asked them to put at the output a little label to say, I see the word run, or I see the word ring, and so on and so forth. And what we found was that we could simulate the acquisition of reading. There are many units in the network that appear that are like the visual word from area. They become completely specialized for reading. And what's great about this is that we can now understand the neural code, because in this artificial model, we can record every single neuron, we can look inside very deeply, every single synapse, and we begin to understand what's going on. There are neurons that become attuned to specific letters, but also specific letters at specific position. For instance, A on the left of the word, A at the beginning of the word. And imagine you have many, many such neurons that care about A at the beginning, A at the end, E at the beginning, E at the end, E in the middle. The combination of all these neurons makes like a little barcode for how we recognize a written word. That's our latest model for what's going on when you read. And I think it's not a bad model for concepts in general. Every single thought that you have is a distributed pattern, a sort of barcode where some neurons are activated, some are inactivated, and the pattern of activation gives you the identity of the thought or the concept. And just a little word of advertising, but I think we'll discuss that also a bit later. This sort of work, where we really understand deeply the mechanics of learning, the circuits that are doing the learning, can lead to an interesting dialogue with the sciences of education. And in the lab, we are doing a lot of work now to translate this understanding of reading into actual helpers for people who teach how to read to children. And we even develop our own software, which is called Kalulu here, and a little reading method that we are deploying in French schools, but especially in Latin America, in Brazil, in Colombia, in Uruguay. I speak about it very freely because it's a non-profit organization. You can download the software and the booklets for free if you'd like. They're available in Spanish, Portuguese, and French for the moment. English is not our priority because there is already pretty good stuff in English, but we will do it in English. So we now have enough understanding, we feel, of the machinery for learning to read that we can actually create protocols, teaching tools that facilitate the acquisition of reading. And in the discussion, maybe we can say more about that. What about mathematics? Mathematics is something that we also do to our brain. We radically change our representations through the acquisition of mathematics. We've discovered that there are areas of the brain that care about mathematics quite specifically. Inside the parietal lobe, which you see here, the back of the brain, dorsal part of the brain here, you can find regions that like all sorts of tools. Manipulations of tools with your hands, but also mental tools, and calculation and numbers is one of them. So you see these areas in red here that respond whenever you do a little calculation. Okay, and they are in the middle of areas that care about space and about tools. Okay, movements in space, movements of the eyes, movements of attention, but also numbers. Very interesting. So if you do 5 plus 2, and you understand the result is 7, you've just activated this area. And you've activated it in a sort of movement-like way, in a sort of spatial manner. That's what we call the number line. You focused on number 5, and then you've moved 2 steps to the right, okay? And you've reached number 7. All of that very quickly, unconsciously to a large extent. But that's what this area is doing. It's putting numbers in a sort of space of numbers, a mental number line. Okay, so we know a lot now about this area, but very recently we started to ask what happens in high-level mathematicians. Do they still use the same brain systems? How does it develop, and so on and so forth? So this is still ongoing. This is my current big research project, so-called ERC project. But this is already an image from a few years ago. We decided to take the brightest mathematicians we could find, ask them some really tough questions in the scanner. So there is a little statement here, a few seconds. It says some, obscure statement in mathematics, and they have to say whether it's true or false. And they have to use their intuition in just a few seconds. And we found that the same network, which in all of us is being used to do simple calculation, in high-level mathematicians is still being used to answer these difficult math questions. You see very strong activation in all of the areas in blue here, including the intraparietal sulcus, whenever we speak about mathematics. When we have questions about algebra, analysis, topology, geometry, this network is kicking in. There is a math network in the brain, and it's quite different from the language network, the semantic network that I was talking about earlier, which is in green here. So if I tell you a question, let's try this. See if you can say whether it's true or false. The sine function is periodical. True or false? True. Many people, I hope, know that it's true. It's not, it's already a little bit advanced mathematics. If I say 2 + 2 is 5, I hope you guys will wake up and say, false. Yes, all right. Okay, now what about if I tell you the taxis in London are yellow? False, right? Okay. It feels very similar, right? I mean, you're answering questions from your knowledge, from your semantics. It feels like you're doing the same thing, but our brain activation shows that you are doing very, very different things. Completely different networks are being activated in the two cases. Right. A math responsive network versus a general knowledge semantics network. Blue versus green here. And of course, if you have not learned this fact, so you know nothing but the sine function, it's all gibberish to you, then you don't activate these areas, right? So it's very interesting that we can begin to map what's being changed in the brain. And now, of course, the name of the game is to see how it is changing in the course of acquisition. But let me show you one more result. So now this is the ventral part of the brain. This is where I was showing you earlier areas that care about faces in red. So mostly in the right hemisphere, a little bit in the left here. Areas that care about houses in yellow, midline, places, tools, okay? But what happens to writing, first of all? Where is it? Pink here on the right. This is no longer working, but you can see a little bit of pink, I hope. Here, okay, just a tiny bit of pink. But this experiment was about mathematicians. So we showed them numbers and we showed them expressions such as a squared plus b squared equals a plus b, whatever. A squared plus b squared is 2ab equals a plus b squared, right? Okay, this sort of expression, or integrals and so on. It turns out that mathematicians, which are on the left here, have an expanded representation of numbers and an expanded representation of equations, okay? And that takes some space in the brain, just like learning to read takes some space in the brain and expands this bit of cortex that responds to letter strings. You see this expansion of these responses to mathematical expressions. So we see it here, you see this almost doubling of the response to the very same visual stimulus when you have become a mathematician and you have a stronger representation of what it means. Now, I have a question for you. Well, I hope I didn't give it away. In what I'm showing you here, mathematicians had more activation. They had more activation to numbers, more activation to mathematical expressions. That's normal because they're experts in these domains, right? But can you guess which of all these stimuli would have reduced activation in mathematicians? Faces. That was an incidental finding. We didn't expect that. But it turns out that the face system is shrunk, has less activation in our images of mathematicians compared to professors of other disciplines. And it's a bit of a funny finding because we all know mathematicians, right? Sorry, I shouldn't say, I'm a mathematician, so I was not so, well, okay. I have many mathematician jokes, maybe I shouldn't tell them, but we have a stereotype of the mathematician and maybe there is a small relation. I should hasten to say here that we don't have any behavioral correlates of this. The brain is flexible. The location of things in the brain can change a little bit. it without any behavioral consequences as far as we can see so far. So far, we have not seen behavioral consequences of this, but expertise in the brain, you might remember this, expertise in the brain changes the relative surface which is allocated to different domains. Okay, back to our initial image. Have we decoded the brain? Well, mostly what I've shown you so far is how we can map the brain by presenting stimuli and showing where it happens in the brain, right? But this image is really about the contrary, decoding the brain to figure out what the person was thinking. Can we revert the process? All that I showed you is based on correlation, and correlation works in both ways, right? So if we are able to see where in the brain we get activation to stimuli, maybe if we see where in the brain the activation is occurring, we could guess what is the stimulus that the person has seen. We could decode what the person has seen. And so this is the very first experiment that was done on this topic, was done by Bertrand Thirion and myself some years ago, 2006, 20 years ago. It's a very simple experiment. In the back of the brain, there are maps of the retina. So whenever you put some spot of light on the retina, there is a little bit of the cortex which gets activated in correspondence to it, actually crossed like this. Okay. So every bit of the retina corresponds to a point on the cortex in these visual maps. So we thought we could reverse it, and that worked. So we can begin by mapping these areas by having rings that expand like this, and they will map out different areas of the back of the brain. And then we can also have a rotating wedge that creates a different coordinate, a second coordinate of this polar coordinate system in the back of the brain. And then we can take any new activity map, and each of these little activation corresponds to a splotch of light on the original image. So we can reconstruct by decoding the original image. And on the bottom, you see that it works. These are not images of actual stimuli. These are images of reconstructed images from the brain activation. We can reconstruct what the person was seeing. And I hope you can see that we reconstruct the expanding circle, or we reconstruct the rotating wedge pretty well here. Okay. So from brain activation, you can reconstruct what the person was seeing. Okay. And in fact, we could also reconstruct what the person was imagining. So if the person was imagining a T, we got a sort of vague image of a letter T, different from when she was imagining a letter O. Beginning of decoding. 2006. Fast forward to 2019, with this gorgeous paper by Kamitani and collaborators. On top, you see the stimulus. On the bottom, you see the image that was reconstructed in part by AI from the brain activity. Here, you're not just using the visual map where in the retina this is occurring, but you're also using all that you know about other brain areas. There are brain areas that care about color. There are brain areas that care about faces. There are brain areas that care about little shapes. So you're using all of this knowledge to try to make the best possible picture of what the subject was seeing. And this is pretty good, right? This is a stained glass panel, and you can sort of see the stained glass panel in the reconstruction here, or maybe some kind of object here. And they made the cover of the journal Neuron, and they made this letters neuron that the subject was seeing, and they reconstructed from the brain activity. Okay, 2019, just to give you an idea of the pace of these things. 2023, okay. And now this is from the work of Van Roelen and collaborators in Toulouse. And see what it's like. This is the test image the subject is actually seeing, and this is for 4 different people, and on average, the image that you could reconstruct using AI from brain activity. This is extraordinary. There's a sink in some bathroom in the actual image. There's always a sink in a bathroom in roughly the same position in the reconstructed image. There are 2 cows in the original image. There's 2 cows, sort of funny, in the reconstructed image in most cases, right? You could take brain activity, and with enough training, of course you need enough training of a particular model for this particular brain, you can decode what the person was seeing. Okay. Surely you'll have questions about that. I want to say one thing. This only works because the person is collaborating. with the experimenter very strongly, spending hours in the scanner without moving, without thinking of anything else than those particular pictures. So don't worry. You're not going to go through the airport and someone will scan you in 1 second and know your thoughts and your code or something. No, this is really heavily experimental, heavily dependent on the ability of the subject to collaborate inside an MRI of several tons, right? It's really difficult to do that in other conditions. But it shows you that there is a mapping. There is a systematic mapping between thoughts and visions and images and brain activity, and we can begin to decode it. Same can be done with other machines. I told you about EEG, recordings of the electroencephalogram. This is MEG, another machine that records magnetoencephalography signals, magnetic signals from the brain. And with that, we can actually decode pictures as well. This is work that we did a while ago. In this case, what's interesting is the temporal dynamics. We can bombard the subject with pictures. Some of them he does not even see. And we can follow in time, this is at a scale of milliseconds here, each and every one of these pictures and decode whether the subject was seeing a face or a house or an object and so on. So each of these diagonals here, I don't have time to explain, but is decoding of a particular category of objects, and we can decode them. And the next picture is really from the beautiful work of Jean-Rémy King from Paris as well. And he's able to decode speech. And this has become a sort of, landmark frontier for brain decoding, which is also explored very beautifully by Eddie Chang in San Francisco, decoding speech so that people who may be paralyzed, may no longer be able to speak, we might give them a new ability to speak, right? So in this case, just from MEG, I didn't think this would be possible. There's 1,000 possible words to choose from, but from MEG signals on a single trial basis, in many subjects, the first word that comes up is the one that the person was hearing in this case. So she was hearing, "Thank you for coming, Ed." And that's what the decoder has been producing in 2 of these subjects. Okay. So this is the frontier. Decoding, finding the signals in the noise, understanding what they mean, it's becoming possible. And one more frontier. If you can read from the brain, could you also write in the brain? Right? Could you have a sort of bidirectional interface? So everybody thinks that's what Elon Musk is doing, Actually, we scientists think Elon Musk is actually not that advanced. But there is other work. This is published in Science already some years ago, 5 years ago, from Peter Hoefsema. It's very beautiful. And again, we should not be scared. It's made for therapeutic purposes, but this is in early stages in the monkey. So what's the idea here? The idea is, again, that you have this map in the back of the brain. It's a map of the visual world. And we know for a long time that if you stimulate with a little spot of light, people will say, "Oh, I saw a little light." Okay. And if you stimulate here, they say, "Oh, there was a light there." And if you stimulate another place, they say, "Oh, there was a light there." Okay. So Peter's idea was, let's put a lot of electrodes. He put 1,024. You see all of these electrode arrays here. And he said, can we paint something on the surface of the cortex? So the first thing is he had all of these arrays of electrodes, and for each of them, he asked where is the corresponding location in the monkey's internal space. So he asked the monkey to make a saccade, and all of these green points are one electrode. If you stimulate that electrode, you'll get a little spot of light for the monkey at this location. Then the key experiment. The monkey is fixating, is in darkness. You stimulate 16 electrodes, and you just select these ones for a given trial. And you see they make an O. Okay. And what does the monkey do? The monkey makes a saccade to the letter O that is appearing afterwards. If you paint a T, the monkey makes a saccade to the letter T. You paint an L, the monkey makes a saccade to the letter L. The monkey is seeing something which does not exist. There is no photon. No photon was involved in this experiment. Just electrical stimulation of the cortex was able to create a picture in the monkey's mind. Okay. This is the very beginning. You can see how rudimentary it is. There is only a small portion of the visual field where you can make letters, very rudimentary. But it's coming. And why should we not be scared? Because this is going to be able to restore vision. The plan of Peter Rufsema and José Sahel at the Vision Institute in Paris is to use this sort of techniques to restore vision. They're already coming artificial retinas, but also cortical stimulation in order to restore vision in people who have become blind for peripheral reasons, right? So this is exciting times where we could have new interfaces to the external world. Okay, this is, I think, almost my final slide, and this is a very special one for me because this is actually my brain. And it's actually my brain from 25 years ago, from one of our early scanners. So I have a backup on the computer. It's very nice to have one. It's probably a bit different now. And I want to use it in a bit of Hamlet style, because that's how you feel. You know, go to the scanner and you see your brain for the first time. It's very exciting and thrilling times, a bit scary. In my case, I came out and they showed me the beautiful anatomical picture, and there was a little hole, a black hole like that in the middle of my brain. I said, oh, what's that? Okay, it was very scary. It was a little artifact from the mathematical reconstruction of the picture. It's not real. It was, okay. So anyway, good memories. So, the real philosophical question is, you know, is that it, basically? That's only 1.5 kilograms of matter, very well organized, very sharply organized, but is it really... It's hard to fathom that this could be my entire personality, my entire knowledge, all that I gave you today in this lecture, all that I acquired during these learning years. Does it fit in? Does everything fit in? And I want to say that several of my friends find this deeply upsetting, and maybe you do as well. The idea that there is no soul, there is no autonomous self, that there is this biological machine. Many of them find it completely depressing. They think that it leads to nihilism. They lead to selfishness. There are these ideas in the literature. Okay. So I want to address it for 1 minute, something like that. I think I understand those fears, because these are real questions, right? But after pondering these issues for several years, I have reached the conclusion that it is perfectly possible to be a lucid, happy, and sociable neuronal man. I think we're all neuronal men and women in this room. Neuroscience research is essentially just the new know thyself. That's how we try to know ourselves. And seeking to understand ourselves and each other is just a fundamental human quest. All religions, all philosophies are in search of meaning. And unless we have no respect for human reason, we just cannot turn our backs on the answers that reason gives us, science gives us about our own nature. So I think we just have to accept that our bodies and brains are just a sort of clumsy hodgepodge of disparate cells and circuits. And I would actually like to argue that this is a little liberating act, knowing about our neuronal nature. Forget about the inaccessible longing for perfection, which crushes us with guilt. I was raised from a Catholic family. I don't know how many people here were raised in a Catholic family. Strict. Yeah, guilt. Okay. We are not made in the image of God. We are just, but this just is extraordinary. We are just the lucky inheritors of hundreds of millions of years of the most sophisticated evolutionary learning algorithm, evolution, followed by thousands of years, or more, tens of thousands of years of cultural fine-tuning. We are the result of an extraordinary process, but that's all we are. Accepting our neuronal nature is a lesson in humility, in tolerance, and in humor. Humility because we do not decide on our neuronal balance. With just a little stroke, a little brain injury, pharmacological imbalance, we can all sink into depression. 1/3 of us will sink into depression at what time during their lives. Schizophrenia, dementia. Tolerance, because the burdens that affect others now might as well become ours in the next years. And humor, I think humor is the only solution, because considered in the eye of a naturalist, human behavior is an inexhaustible source of amusement. And I hope you'll think about it during your child's next tantrum, perhaps. So with these mini philosophical thoughts, I would like to thank you, and I'd like to add one touch of humor. This is my wife and I wearing our equipment in the lab, as we do sometimes, and having fun exploring the brain. Thank you very much for your attention.
Thank you so much for this amazing lecture. I can say that this applause was like really, really long. So in comparison to other events. So thank you so much. Thank you so much for coming. And now, sadly, we don't have time for questions, but this is not the end for today. So now we would have 15 minutes break. We would reconvene here 8:15 for the panel discussion, and there would be opportunities to ask some questions to Professor Dean during this panel. So stay with us, get some fresh air, and we see each other in 15 minutes here. Thank you so much again.